Good morning, everybody. I am Gabriel Chambers,
My name's Peyton Blake.
My name is Sharon Iweha.
My name is Oluwasegun Soji-John.
and we're at Project One. We'll be diagnosing and mitigating modality dominance and multi-omic antidepressant response predictions, and looking at how can we improve our AI predictions for antidepressant responses. Okay, so going off the background, just multi-omic learning combines different or just multiple biological data sources to improve disease prediction. And normally, we're coming with a traditional assumption that more data means better predictive responses or better predictive performances in our models. And there has been research that shows that adding additional modalities does not always improve prediction accuracy. Instead, there are very high dimensional but weakly informative modalities, meaning weaker signals that can dominate the whole learning process when it comes with model learning and reducing the overall model's performance. So our key question is, how can we identify and reduce the influence of weak biological modalities while preserving the most informative signals? So our research is driven by three core questions, and that is, does modality dominance truly reduce predictive performance in multi-omic learning? We need to determine whether those weak, high-dimensional modalities actually suppresses the important information that we need based off our biological signals, and does it lead to those poor predictions? And is performance degradation driven by biological signal quality, or simply is it because it's just high-dimensional? We need to separate those effects of those biological dimensions and from the dimensional noise using control experiments and synthetic noise substitution. And can adaptable modality weighing improve the prediction of that multi-model as a whole? So we're looking in evaluating our own framework, signal-gated omic fusion, or SGOF for short, and that learns the important signals of each modality to improve an overall model performance. So our hypothesis is models that adaptively weigh biological modalities based on predictive signal will outperform models that treat each modality equally. So this research matters because it challenges the traditional assumption that more modalities always improve prediction, and explains why multi-models, they sometimes fail. And the technical impact is that it introduces an adaptive modality weighing through SGOF and improves overall robustness without removing the important biological data that we need to support more reliable antidepressant responses predictions and moving towards a more curated treatment strategies and precision medicine. And our overall impact will advance the reliable, interpretable, and patient-centered AI models, precision medicine.
So from our lit review, we learned 6 things: what we know and what was missing when it came to multi-omic research. First, we learned that multi-omic data improves predictive models. So in our case, our multi-omic layers are gene expression, genomics, and DNA methylation, and how that can improve disease prediction, or in our case, antidepressant responsiveness. Second, we learned that with multi-omic data, you need deep architecture. So in our case, the paper we read focused on graph neural networks and how that can capture complex relationships in between the different multi-omic layers. And third, we learned that feature selection and regularization are commonly used to address overfitting and to reduce high dimensional noise that comes through a lot of data sets. We also saw that a few things was missing when it came to multi-omic research, such as modality dominance being underexplored or not really valued when it came to the research. So high dimensional but weak signal modalities and how they affect high informative signals wasn't really something that was addressed throughout our research when we saw our papers. Second, we saw that signal versus dimensionality wasn't separated. When it came to signals, or when it came to models that did not perform the best, they would just say it didn't work. It wouldn't tell you why, whether it was because a data set had a low signal or whether it was because of high noise due to the dimensionality of certain data sets. And lastly, there was a lack of adaptive weighting mechanisms. So like Gabrielle mentioned, there's an assumption when it comes to multi-omic data that every layer contributes the same equally with its information, but that's simply not true. So our research gap was addressing all 3 of these things. So how do we do that? First, we diagnose modality dominance by looking at how high dimensional noise hinders the predictive performance of our predictive models. Second, we look at the source of the collapse. We don't just say it doesn't work, we figure out the why. We look at whether it was due to weak biological signals or high dimensional noise. And lastly, we propose SGOF, or signal-gated omic fusion, which is an adaptive framework that prioritizes the importance of the modality over fusion. So this is a visualization of our workflow. Like I said, we have our multi-omic layers, genotype, gene expression, and DNA methylation. And instead of just putting all that data together, instead we put it in our graph convolutional network encoder, or GNC encoder, so we can look at the important representations of the data while simultaneously keeping it contained to its individualistic layer. And then from there, we put it into our SGOF weighting by putting it in a held out validation, so we can predict the estimate, so we can estimate the predictive signals of each modality. And from there, we get to our final classification between our respondents and our non-respondents. So in our data set, we have 111 patients that have MDD or major depressive disorder. 61 of them are respondent to the antidepressant treatment, and 50 of them are not. So we have our 3 multi-omic layers: genotype, gene expression, DNA methylation, and our pre-processing pipeline. So we aligned our patients based on their modality. We did mean imputation. We used ANOVA to select the top 200 features of the modalities. We used our Z-score standardization and model input. Now, comparing to current models and what most typically they use conventionally, there's early fusion, late fusion, similarity network fusion, or SNF, XGBoost, MogoNet, and MoGCN, but one stood above the rest, and that's SGOF, because it prioritizes the importance of the modality above all else.
Yeah, so just to continue on what my colleague has already started on, for methodology, we started with tenfold stratified cluster validation, meaning we did 10 folds, trying to keep the same responder to non-responder ratio in each fold. And we held out 1 fold, meaning in each training set would have 10, but would hold 1 out, so 9, and keeping the ratio of responder to non-responder the same across all the folds. And we also did a lot of imputation for the missing values that existed in our data set. We proceeded to train the baseline models, and these models are basically like fusion families, conventional methods by which different types of modalities are combined together to perform, in our case, to predict whether or not patients would respond to antidepressants, patients with depression. And then we proceeded to train our framework, and we did some performance comparisons between the fusion models or baseline models and what we've currently developed. Yes, and for the metric of evaluation that we used, these are the ones that we considered. And amongst all of these ones, the one that we prioritized is AUROC, which kind of measures the ability of the model to classify whether or not a patient is a responder or not a responder. And for AUROC, we have like a threshold of 0.5. And when you have a score that's higher than 0.5, it means it's above chance, meaning that the model is not just guessing randomly, and it's actually giving predictive contributions that make sense, right? So the closer you have above 0.5 and closer to 1, the better the score is for that particular model. And we also consider things like PRAUC, BACC, and ACC, but we prioritized AUROC out of these methods for evaluating our model. And for the key findings from all of the technical work that we did, the first one is high dimensional imbalance. And basically, this is high dimensional data is, let's say in our data set, for example, we have 111 patients, meaning we have 111 rows. And then for the genotype layer, I think our teammate showed it's like 250 something thousand features, right? So that's very highly dimensional. And that imbalance can affect the performance of fusion when you join these different modalities together to perform, right? And for gene expression, 1,572, and DNA methylation, 13,900. And you can see from the one on the rightmost side the responder to responder ratio, and the similarity between how the genotype layer was classifying patients. You want the layer to be able to carry enough predictive signal so that each patient category, whether they're a responder or not, can be significantly classified. But when there's so much similarity and there is not enough signal, you can see that it's not carrying what you need from it. Yeah, so for the other key finding, I know that's a couple of figures, but these are just the baseline models that we did, or fusion families, conventional ways of joining different types of biological data together. And from these, you can see that in the green is when we've removed that genotype layer, which I said was very high dimensional. And on the purple for each of the models, you can see that it's when the genotype layer is included, right? And for most of these, you observe a trend, which is when you take out that layer, it gets better AURC scores, meaning it has better signal and it can perform better in terms of its responses on predicting whether or not a patient will respond to antidepressants. And for all of these, SGOF is the one on the right, and you can see it doesn't have the highest score. And that's an important thing. It doesn't have the highest score. The highest score here is late fusion. But what it does is it's the most robust in the sense that it has the least amount of difference between having that non-informative layer and having it not present, right? So that shows that even if you were to add noise, a layer that has a lot of noise to the data set in Fusion, it would still perform good because it's able to do what it's able to generalize. And that's a very important thing that is considered in joining different types of biological data together. Now to the next slide. Yes, like I said, late fusion had the highest AUROC score, and that just shows that on this slide. And for the next, so the framework we did something called adaptive weighting, where we kind of assign weights to each modality or each layer of data. We have 3 layers of data in our project, DNA methylation, gene expression, and the high dimensional one, which is genotype. And by assigning weights based on the amount of predictive signal that each of these layers contributes, we're able to kind of reduce the influence of a layer that has a lot of noise, even if it's not adding to the predictive contribution, right? So here we can see that the gene expression layer has the highest weight, DNA methylation has the second, and then genotype has the lowest weight. And that's because it carries the least signal, meaning it doesn't contribute to the classification of whether or not a patient would respond to antidepressants or not. And for the interpretation of the results that I've been discussing, the first thing is that high dimensional data can be misleading. In a lot of research that's conducted, there's this conventional thinking that the more data you're able to add different types of biological data to try to figure something out or try to train a model, the better it would be, right? But that is not always the case, and that's what we've been able to show. And equal weight fusion is basically thinking that each layer of data has the same amount of signal that would help for the model to classify in whatever case you are doing, right? And also adaptive weighting captures what matters, because when you notice that something doesn't contribute as much as the other, it's able to give it a lower weight, and by that, it's able to get better performance after fusion. And also stronger models support better decisions. We can see the SGOF adaptive weighting, just a quick display of what that would look like. And yes, so that's the interpretation of our results.
In addition to wanting to build an accurate model, we wanted to make sure that it was also responsible and trustworthy. So for our bias and fairness, we wanted to make sure that all pre-processing was performed using only the training data and within each fold to prevent any type of data leakage. And for SGOF, it prioritizes the information, the more informative data over trying to make everything equal. So with that, it helps with limiting the noise that could interfere with the predictions. And for our privacy and security, we made sure that all patients were de-identified. So we never mentioned names or age or anything like that in our data. and our data was within controlled access to make sure that it didn't get into the wrong hands. And also our we reported all our findings aggregately to make sure that there was no leakage, and to make sure that all our cross validation statistics were based on an individual basis. For our limitations and our future work, our first and major limitation was our sample size. We only had 111 patients, which made the generalizability and also the scalability more complicated. So it would be harder to base our data or use the model for larger populations. For our data was also very high dimensional, which made it a little bit more complicated to analyze. And it also required a lot of cross validation. And for our future work, we would like to test our model on different, more diverse data sets, and also see it used in real world clinical settings to improve robustness and clinical utility. And would also like to improve our model architecture as well. And to conclude, our model actually shows that choosing important data over making everything equal helps with the predictive performance, and it also helps with making the model more clear and interpreting the results better. And our model uses SGOF, which helps with weighing the modality weights from the data, and also it helps with reducing noise and increasing robustness. And one key takeaway from this is real data in our case outweighs more data. We would like to acknowledge our faculty mentor, Dr. Jamell Dacon, Ricky Gole, our graduate mentor, and SAIRI and CEAMLS program as a whole. These are our references, and I would like to open the floor for any questions.
We have about 5 minutes for questions from the audience. Floor is open.
You guys spoke on the fact that you have 3 layers in your, I guess, what is the importance of those?
So the importance of those 3 layers is gene expression captures what a particular gene does, and DNA methylation is basically it's like a regulatory something that works with whether or not a gene is active. That's what that controls. And then genotype is basically just capturing genes that are related to your genotype. So they carry different types of biological information. And in our lit reviews, we saw that these were things that people would do in terms of looking for whether or not depression is in someone's genes, right? And that was why we chose to use these layers.
How do you account for false positives, when you're making these predictions?
In accounting for that, I think a majority of it was just like testing it with other models and seeing whether or not we were having like a score that was crazy high or imbalanced with other ones. I think that was the major way we would look forward to like trying to identify whether or not that was happening.
And I was also going to account for also our tenfold method. With a lot of cross-validation, it does stop us from having those false positives.
Why do you think the
Just like Shagun had mentioned before, it is very high dimensional. So therefore, you would think in the case of this, because it's so high dimensional, it would have more data, but it was so much noise in that data and it carried that weaker signal. So therefore, taking it out, we would have a better signal overall for our model prediction.
And just to add to that, it had the highest amount of features. You saw it was like 200 something thousand. And at the start of the project, our thinking was that would be probably like our best performing layer, basically because of how much information was contained in it. And as we progressed, we're able to like identify that that's not the case and work with how can we mitigate that in terms of predicting whether or not a patient would respond to antidepressants, right? So that was how we came up with like the SGOF framework to mitigate that cause while we still have a model that would predict what we want at the end of the day.
I have a question. So you said you guys use the segregated thing. How is that different from traditional medical?
So that's different because we're doing something called adaptive weighting, which is simple at its core, but it's very effective, meaning for usual fusion families, they have different ways that they fuse things together. For example, early fusion is you join all the different layers together. Initially, you join them together, you have this one big concatenated data set, and then you proceed with your testing and validation and all that. And then for late fusion is you train on each of the layers separately, and at the end, you get the results and you average it out. That's late fusion. But then with SGOF, it's like you give weights to each of the layers. And after you give those weights, you're able to know which layer or which layer carries signal, basically which layer can actually give good contribution and which layer does not carry signal. So it's pretty basic at its core, but it's very effective in terms of mitigating high dimensional data or noise in the fusion. Yeah.
Do you have any difference with or weird?
I would say actually it goes back to what I was saying earlier. We found out that that layer was not adding good contribution to our fusion. I know I've said it all the time, but that's literally what it was. So in that case, I think that would be when we realized that yeah, it wasn't really learning enough because the scores were lower. Observe it like we could observe like across all the baseline models that we tested initially that the scores were lower because of the noisy layer that we had.
Yeah, you can see it on the board. Well, it's on your side, but yeah, it was a big difference. And yeah, it was a very big difference. Did I answer your question? Okay.